So welcome back to our lecture series 2013-2014, and we're really looking forward to our scheduled list of speakers this season. To start off, the beginning of the seminar series, we have a visitor from University of Windsor. Dr. Douglas Haffner is a Tier 1 Canada Research Chair at the Great Lakes Institute for Environmental Research. Dr. Haffner's research interests include contaminant and energy flow dynamics and aquatic food webs, processes regulating speciation in ancient lakes, and more recently on the effect of non-point source pollution on the Three Gorges Reservoir. He's a president of the Society for Speciation in Ancient Lakes and co-director of the China-Canada Three Gorges Reservoir Water Science Center. Very large name. So what you may not know about Dr. Haffner is because we like to share a little story or a little bit of knowledge or fact about them, is that when Doug was in the north, he was along the Polar Bear Pass, and he thought he could walk across the ocean in just one day. But in fact, it took 3 days. Because it was so clear and he could see the icebergs, he really had no idea what he was in for, and he almost froze. So this evening, Dr. Haffner will be sharing with us his knowledge on the Malili Lakes of Sulawesi Island. And following his presentation, we will have some time for some questions from you guys. So if you could please help me welcome Dr. Haffner.
Thank you very much, Kate. It's a pleasure to be here. Yes, after my experience in the north and freezing to death up there, I decided I'd work in the tropics, and quickly headed off down to the south. What I'd like to talk about today, basically, is a kind of like a mid-career change for me, kind of like an academic menopause, perhaps. Because I basically had been working in ecotoxicology along with Chris Metcalf in this, but I had the opportunity to actually go to Indonesia and provide some courses on hazardous lakes because of Lake Kivu's explosion in Africa. And the question was there any kind of lakes like that in Indonesia? And after the course, they said, where do you want to go? And I said Sulawesi Island, because all my life I've been in love with Sulawesi Island because of Alfred Wallace. And I didn't really know in those days as to what I was getting into when I arrived on Sulawesi Island overall. Basically, I'm going to start this off by talking about a famous letter that was mailed actually March 10th, 1858. And it was mailed by Alfred Wallace, the man on the left here. And in this letter, he includes a manuscript. And he sent the letter and the manuscript to Charles Darwin, asking Charles, please read this. If you think it's any good, would you please submit it to a journal? Because obviously he was stuck down in Indonesia and had no access to submitting his work to any kind of journal. The journal, or the manuscript, obviously shocked Darwin, who went into almost a hysterics, because it basically had scooped him completely on his concepts of natural selection. And oddly enough, the letter that came with this manuscript, Darwin was famous for being really anal. He could come up with anything he collected, any letter, any correspondence he ever had, but he lost this one letter. And everybody has always wondered what was in the letter from Wallace to Darwin. And of course, what happened afterwards is on July 1st, 1858, there was two famous manuscripts read at the Royal Society. One to be read by Charles Darwin, who lived just down the street and didn't show up, and the other by Alfred Wallace, who was obviously in Indonesia and couldn't be there. And so it's always left a question as to who really is the father of evolution. And everybody has always wondered ever since what was in that letter from Ternate. And what I'd like to talk about basically is, at least from the quotes from the manuscript that was sent to Darwin, is Wallace talked about basically descent from the original form, or modification from the original form. And you can see why this happened when he was down here in Indonesia. And at the time, he made a very famous trip, basically between Bali and Lombok. In Bali, basically recognized the flora fauna consisted of Asian animals, and plants, which actually had an origin with some sort of ancestor in Asia. But as soon as he went to Lombok, he recognized the original sort of parent or ancestor was going to be Australian. And he basically, after that, went and looked at the different islands of Sulawesi, trying to figure out which island had an Asian element or an Australian element. And he even hypothesized, and quite correctly, there must have been land bridges. He actually recognized that, the water levels must have been different. Now, he actually thought the land levels rose, as opposed to what we know now, that in the glacial periods, the water levels collapsed, joining many of the islands, creating bridges for animals, plants to move across overall. And so this basically was giving him the format as what he wanted to talk about, and it basically became known as Wallace's line. He basically drew a line between those islands which were influenced by an Asian influence and those islands that were influenced by the Australian influence. But what he couldn't figure out was this crazy K-looking island here called Celebes. Now, Celebes is Portuguese for basically the island of the infamous ones. And the infamous ones were the people from Makassar, who in those days were called boogies. And that's where the expression, watch out, the boogeyman will get you. They were famous pirates. But basically Wallace was able to work with these people, sail with these people, but he kept working on Celebes or in Sulawesi because it just didn't fit in terms of either an Asian origin or an Australian origin. And he was over here, basically on Ternate, when he wrote his famous letter, because he actually had to leave Sulawesi. He actually had a nice case of shingles and malaria at the same time. If anybody's had shingles, you can think how bad it was. Add malaria to that, and you can understand what Alfred Wallace was feeling like at the time. But he basically spent a lot of his time, he wrote a book basically called The Ultimate Island, Island Life, and he referred to it as the anonymous island, but he just couldn't really figure Sulawesi out. Even before he died, he was suggesting perhaps his line Wallacea should be off to the east side here, but it always puzzled him as to what was going on Sulawesi Island. Well, to understand Sulawesi Island, we have to understand basically perhaps its origin. So what I'd like to talk about today is about three kind of sectors of my talk. One is to talk about Sulawesi as the ultimate island, also talk about these ancient lakes that are on Sulawesi and why they're so important, and why these lakes are going to become extremely important in our near future as we start understanding drivers of climate change on our planet today. So basically you can see Sulawesi Island sits here in an area of a large amount of tectonic activity. Major plates are moving about overall. We have the movement of the Indian-Australian plate coming up from the south. We have the Caroline plate coming off from the east here, kind of a static Eurasian plate. But basically on Sulawesi, the whole island is being twisted in a counterclockwise motion, and it's splitting apart, it's fragmenting, and of course forming major fault lines. And some of these major fault lines basically show up here, and these lakes I'm going to talk about are the Malili Lakes overall. And basically the major fractures have started to occur. One is the Palu-Koro Fault. Back in 1996, the city of Palu was almost ruined. And basically the other fault is known as the Matano Fault, which is the lake which actually kind of grabbed my interest for the past 20 years of my life overall. And you can see the motions of these plates overall. And you can also see in this area some very deep waters surrounding the island overall. So when the water levels were at low, Sulawesi remained an island. There was some conjecture, perhaps at one time during its formation, it may have been pushed up against Borneo. But the data we have right now suggests not. Actually, a lot of that data originated here at McMaster with Ben Evans' work on Sulawesi Island. So it's kind of nice to be here, having somebody who's worked on the terrestrial system and somebody who's worked on the aquatic system, kind of a bookends approach to Sulawesi overall. And the other aspect of Sulawesi, you start seeing the North Sulawesi Trench and the Molucca Sea. So basically, as Wallace was proposing, it's an island that has always been an island. It's never had a connection to any other major landmass. And what's ever happened there has happened since its development overall. And, the Philippine Trench and these. So you can sort of get an idea as to what's happening on Sulawesi Island. A lot of movements occurring on the system. These are representing earthquakes in the area. It's quite a lot of fun doing limnology in a high earthquake zone. Actually being on a lake is quite safe until you start thinking about, there's a dam down in the end here, and that goes. Maybe fast sailing. I'm going to show here basically the formation of Sulawesi Island. Keep your eye on this arm here. This is the volcanic arch that you see sort of stretching off on the top of the K. And we're going back 22 million, 21 million years, and we can gradually see through plate tectonics the formation of Sulawesi Island. So it's actually not a single island, but it's basically a series of islands that have come together overall and have been islands for long periods of time. So we're basically getting 5 million years ago, and you see the island starting to take shape. 2 million is when the lakes are starting to form, and basically we have Sulawesi as it is today. So when you look at it geologically, Sulawesi is a really complex but very wonderful system to work on. You have a large volcanic ridge down to some calcareous rocks. You have these ultramafic rocks, high lateritic soils, and other sorts of rock geological structures just off on the east arm here overall. So it's an island, and we tend to think in terms of the how organisms got there are two ways. One is by carrions, basically moving along with the actual other island fragments, or by dispersal and landing onto the island itself. And there's an excellent paper that just came out on that topic. Moving towards the lakes themselves, this gives you an idea of the geologist's view of the Lake Matano. This is Lake Mahalo, Lake Tawuti. These make up the Malili Lakes. And this shows you the Matano Fault line overall. It was geologically, so the geologists were saying at one time, that this was one lake. And the data sets I'd like to present you today are biological data sets that says the geologists are wrong. I'm going to try to convince you that actually these are three separate systems that have actually come together over time, forming a rather neat evolutionary laboratory for us to study in terms of understanding speciation and biodiversity overall. This is a radar image of Sulawesi overall. And again, you can see the Matano Fault running along through here. If you look back a bit, up the top here, you may see what may be a paleo Lake Matano. Now, Lake Matano itself right now is about 2 million years old, but there may be some evidence that Lake Matano itself is just moving down the fault and is actually making recent contact with these other lakes which are in this more complex sort of faulting area overall. And if you sort of look at the lakes and figuring how old they are, they fit there among some of the better known ancient lakes of the world. From Lake Victoria, about less than a million years old. Lake Titicaca, 2 to 3 million years old. Of course, sort of the grandfather of them all is Lake Baikal, 25 to 30 million years old. And of course, over here we have, another lake on Sulawesi Island, ancient lake, Lake Poso, about 2 million years old. And then the Malili Lakes, where we figure Matano is about 1 to 2 million years old. Tawuti, we've got some good evidence, is close to about 1 million years old overall. But the neat thing about the Malili Lakes, they're the only ancient lakes in the world with a hydrological connection. All the other ancient lakes are individual systems sitting in their own basins. And, so basically when we talk about endemics and how endemics can move or can't move, we sort of say, well, they're geologically isolated into that system. But in this system, we would expect to see a fair amount of dynamic interaction amongst the formation of endemic species flocks that could be forming in the Malili Lakes overall. Yeah, when you look at the outfall of Lake Matano, beautiful area, isn't it? Like, wouldn't you love to work there? Gorgeous beaches, the water, we have a secchi depth, like one of those little pie plates, goes down about 25 meters, and that's because my eyes can't see any further than that. But we actually have measured light down to 100 meters in Lake Matano. It's a super clear lake overall. But if you look at this drainage system, this is the Pethia River. It certainly doesn't look like something that's 1 million years old. There'd be a much deeper cavern. There's a rather fast-flowing system. And so we tend to think that it's only been more recently that these lake systems have hooked themselves up together and had some kind of connection. Now, in the past 20 years since we started working on the lakes, they really become quite famous because of their endemic species flocks. And these were first recognized back in the late 1800s, because actually Wallace didn't work on lakes. And it really wasn't until the Paul and Fritz Saracen came in 1893-96 and came into the area, and they were basically interested in looking at the biology and endemic species, and especially in terms of Lake Poso, shown where it is, and the Malili Lakes shown there. And they were collecting basically snails, and they recognized right away these were all endemic species. And they got quite excited, and they went down to the Malili Lakes and found another whole suite of endemic snails, and of course returned again in 1901, 1903 to do their collections. In 1930, there was the Woltereckii expedition who came into the area, a German expedition, and they actually sampled diatoms and found again, basically all the diatoms were endemic to the lakes. Now this surprised the diatomologist those days was a guy named Husted, who was kind of the father of diatom taxonomy. And of course he was quite thinking that once you study diatoms of any one lake, you could do diatoms in any lake in the world. Like if you studied diatoms in Lake Ontario, you could fly down to Australia and start identifying diatoms down there. So it really amazed Husted when he started looking at these Malili Lakes as to all these endemics of something that's supposed to be cosmopolitan, yet the lakes are filled with endemic species. Basically, the Malili Lakes have about 99% of the species are endemic to the lake itself. The next highest is Lake Tanganyika at 50%. And actually they have more endemics, but what they do have also in Tanganyika are cosmopolitan species. These Malili Lakes really don't harbor any of these other more cosmopolitan species. They maintain their own niches are filled with their own endemic species overall. So it makes it a wonderful evolutionary study system overall. It's a really neat system. We have endemic fish populations. There's 4 endemic generations. There's the Silphin silversides, Telmathenids, the half-beaks, the hemirampids. I guess everybody has gobies, but these are endemic to the system, and actually even an endemic rice fish. We also have endemic crabs in the system, endemic shrimps to the system. Snails, a lot of interesting work done in the snails after the Saracens' work. And quite interesting enough, really a nice variety of diversity of endemic sponges, which is really rare to find in any of the other sort of ancient lakes that you might want to go study overall. There's been a fair amount of work done since we started working there in terms of the number of species. An interesting number of fights too as to one thing I found out very quickly with people doing molecular biology, they never agree with each other, especially if they're studying fish. There's something about molecular biology and fish that there's just complete disagreement. Like you even have people sort of claiming this is my lake and don't you work on my lake on that. But I can actually say this is a pretty open laboratory. If anybody's interested in coming down and working with us on this system, you're more than welcome. But we do have battles as to number of species. I completely disagree with the number of silversides at 31, and I'll show you why in a little bit. But basically, you get an idea that, yeah, we have a good number of species, but you can sort of see already it's not species rich. A lot of them are endemic, but we don't have that species richness that one would associate with old lakes that are 2 million years old overall. It's really a neat system. If you're into looking at shrimp, these are the types of shrimp species that we actually find. This work was done by Thomas von Rittelen. And of course, after he produced this paper, we got inundated with people from pet shops that were out there to collect all these endemics and take them back and sell them to people for like $4 or $5 each type thing. But also caused another problem. It's a serious problem too, especially I think in terms we talk about United Nations University, is when that happened, it changed the permit system in terms of doing research and being able to have collectors' items, and being able to take specimens and study specimens someplace else. It really changed our world dramatically as to how we could move forward with our research within collaboration with the Indonesians. Interestingly enough, the riverine species are basically pretty bland. It's only in the lakes that we actually see this diversity of color being evolved overall. And as I say, this was the kind of stuff that resulted in our being inundated by collectors. Of course, Alfred Wallace was a collector. That's really why he went down to Indonesia. Like he was after the bird of paradise. That's how he made a living. He was out there shooting things, collecting butterflies or whatever it might be, and selling them back in England It's just that he happened to make some really neat observations on the way. So basically, looking at the Malili Lakes as a system, we can see here Lake Matano. So it's 600 meters deep. It's one of the deepest lakes in the world. Lake Mahalona, Lake Tawuti, Lake Lawantoa, and Lake Masapi. Masapi means eel, and there's a lot of eels there. But these are the basic lakes, and you can see basically the hydrological connections that are among these lake systems overall. So we expect to start seeing gene flow. We start seeing some endemics were in Matano. Those endemics should move downstream into Mahalona, should move downstream into Lake Tawuti. Same be true from any endemics from Lake Moatoa. They should be able to move downstream. And so we were quite surprised when we first started our studies on the system to start seeing these kinds of patterns. Is that basically shown here are the snails, shrimps, crabs, and fish, the number of species in each lake. Again, you can sort of see, low species richness overall in the system, and whether you're looking at snails or shrimps or fish or not. But what gets really neat is when you look at the number of endemics. 10 out of 10 fish species in Lake Matano are endemic to Matano. And basically, 2 out of 7 in Lake Mahalona, 7 out of 12 in Lake Tawuti. And if you look at an overall aspect in terms of the snails, greater than 75% of the endemic snails are in one lake. Even though we know the propagules can move from lake to lake because of hydrological connections, because of animals moving back and forth like birds or something like this, we know those animals and can move about. And we think about the invasion meltdown theories of Simberloff and these people would sort of say if a propagule can get there, we should have a great homogenization of our species in the system. Well, here's a system that's 2 million years old. It has propagule pressure. Things are moving, but it's maintained very unique community structures of these endemic communities. So maybe we shouldn't be as afraid, perhaps, of the meltdown theory as it's been proposed to be. There truly does seem to be such thing as ecological resistance, which has been a concept of Odom that's been shot down for a long time, and I like to see it kind of brought back up again, that there truly is such thing as ecological resistance in these systems. This is the same true in terms of looking at the snail populations of the systems. We tend to have different types of snails living on different types of substrates, and I'll get into more detail in that. But I'll first start with looking at diatoms. As I said, we've got about 250 species of diatoms we've found so far in the Malili Lakes overall. Now in the Detroit River, got 1,000. That gives you an idea. We don't have the species richness, but almost all of them, except for 4, are endemic to the actual lakes themselves. You will not find these diatoms in any other lake system. What is also interesting is the number of endemic species. And look at the number you tend to find in just one lake. Out of 250 species, 140 diatoms are found only in one lake. Now, somebody like Ralph Daly, who's worked on lakes, would probably tell you that's quite astounding to sort of see something where diatoms themselves are not moving from lake to lake. That you can basically sample diatoms in one lake and go across the road and sample and find the same community. Not here. So basically, in terms of all these 5 lakes, only 4 species are found in all 5 lakes overall. So there's something about the evolutionary processes here that's basically resulting in these endemics basically being unable to move from lake to lake, even though we know they're getting there overall. I'll just skip this, it's too technical. But the same is true in terms of looking at some of the phytoplankton. These are Gymnodinium, looking from Lake Matano. And you can see the species of Gymnodinium in Matano is very different from that in Mahalona, which is very different from that in Lake Tawuti. These things coexist. They have pretty well the same type of seasonality, and yet we know this one flows downstream to Mahalona, but it doesn't survive there. And we know it's in Mahalona, comes down to Tawuti, it doesn't survive there. So there's something really neat going on. The speciation processes that are going on are very unique to the lake systems themselves as to which species are there and the types of endemics that we find in the system. This gives you an idea how beautiful it is if you take a quick swim there. It's just like having a coral reef. These are the sailfins or telmathins, as you can see in this picture. And And we're going to talk a little bit about what's going on in these lakes and looking at it in terms of adaptive radiation. And we're really going to be talking about the take-home message is trophic specialization is involved in almost all the radiations we see in these lakes. So what's ever driving this lake-to-lake differences, but it always seems to be driven by a trophic interaction in terms of how you make a living, where you get your food from, where you're obtaining your energy from overall. And I'm also going to maybe veer away from the traditional ancient lake work, because almost everybody works on either benthic or littoral zones. I'm also going to venture out into the actual pelagic zone, and I'll talk about speciation in a pelagic zone. It's something you don't really hear about in ancient lakes. The cichlids and all that, those stories are always around rocks or substrates of the shores of the lake. Very few people talk about what's going on in the middle of the lake itself. So looking at these cell fins, this is work of Fabian Herder. You can basically sort of see how these have branched off. You've got the Matano fish, and basically there's two clusters of fish up there, the sharp fins, and we call the round fins over there. And then basically it links to Wuti, Mapelona. We have again some of the tomathids. We also have these paranthids. None in Lake Matano. Eels can go up the river, other things can go up the river, but not paranthids. They don't go from the Wuti and Mapelona, and have not ever invaded over 2 million years into Matano, assuming there was a connection at that time. And of course, the streams maintain their own type of telmathid population overall. And these are basically all trophic type feeders. You have fish feeders, egg feeders, mollusk feeders, insect feeders, very specialized feeding structures, what they tend to eat overall. And this started to become part of the fight between us and the German research group. And if you ever fought a German, it's tough, but I can do it. My name's Haffner. And you can see here that, you look at these lakes, these are the telmathids we find, very unique assemblages amongst these lakes. Even though the fish can move about, there's basically 10 out of 10 of the telmathids of Matano are endemic to the system overall. And definitely fish should be able to move downstream. So we started looking at what was going on in some detail as to these fish and looking at these different types of genera overall. And, whoops, go back a bit. There. This is studying fish in the telmathids in Lake Matano. And what we found, just basically using the 16S rDNA, is they would basically cluster out into 3 clades. And so where the Germans were saying there's 10 species, we looked at this and said, we're seeing maybe 3. And, and they said, oh no, you're wrong. And so we went a little bit further and started doing morphological analysis. And we did warp analysis and looking at head structures. And guess what? 3 basic structures which actually correspond to our 3 genetic clades that we found. We started looking at the pharyngeal jaws, and the same type of thing occurred over again. Looking at the jaw structure, it followed the same basic structures that we saw with the 16S DNA studies. And the same again, So you already see resource partitioning. These things have highly specialized. We've even done this work with isotopes and can actually show how they're more of a littoral zone and a pelagic zone type fish. What is interesting when you move downstream, remember I said that paranthid lived only downstream. You could actually go downstream because we know this paranthid doesn't come upstream, and we can actually do the same study again and basically repeat it and come out with the same type of data set. Is that you can see here with the paranthids, which are only in Lake Mapelona, in this case here, you can sort of see how they divide it out, but we got the littoral zone and a pelagic type resource structuring. Those which are actually feeding in the littoral zone, those which feed more in the water column. The same things happening with the telmathids in Lake Matano. A beautiful example of parallel evolution occurring using resource partitioning as the driving factor behind speciation overall. We can see the same thing again actually with the snails. Now this figure didn't come out the way I wanted to, and I just found that out just about 2 hours ago. This is work by Thomas von Rintelen. And he basically did the same thing. You can see basically 3 distinct invasion fronts of the snails. But if you look within the actual systems themselves, you can see either the same process, either adaptation to hard substrate or a lacustrine type taxa occurring over and over again based on the form of the radula, shape of the radula. So we're seeing within these different evolutionary or different colonization time periods where three different events occurred of colonization. After colonization, they've all gone down the same basic route of parallel evolution, adaptation of the radula to feeding either on soft substrate, on wood, or hard substrate overall. So we're seeing the same thing. We see it in fish, we see it in the shrimps, we see it in the snails, in terms of how these invasion fronts move forward, and speciation tends to occur afterwards. So I promised to do something which ancient lake people don't do, is what's going on in the pelagic zone. And the reason you don't touch the pelagic zone, it's homogeneous. You know, you shouldn't be able to get some kind of adaptation to something because it's homogeneous. It's a well-mixed, sort of middle of a lake kind of process going on. And, you know, you're also dealing with a species like zooplankton, which have a high dispersal ability. So there's very few barriers to dispersal. And we're asking a question ourselves behind this, is how important is hybridization? And of course, this is also an excuse for me to do a trans-Sulawesi trek. I'll have to admit that there's a non-scientific reason behind this, is because it's the most fascinating thing you can do in your life is go to Manado and work your way down all the way to Makassar. It's just, you're going through so many different worlds, so many different cultures. It's an incredible trip. You know, it's a lifetime trip. You'll remember it for the rest of your life. But there's some neat lakes in the system. Lake Ternate up near the top part, north part of the island. Lake Pozo, which is in the middle part of the island here, and the Malili Lakes. And what we did basically was ask the question, well, you know, what are the zooplankton like? Was there basically just one basic founder of zooplankton populations in these different lakes? Or did they have different founder populations overall? So we went through a process of morphological identifications and DNA sequencing and, you know, the typical stuff. Doing both mitochondrial, CO1, and nuclear genomes in this case. So looking at Lake Ternate, first of all, you can sort of see here that we have a singular nuclear genome. But look here in the CO1. You know, we actually have quite two different clades that are showing up there overall, with about a 15% divergence. Now, if you were a barcoder, you'd say, oh, there's two species there. And when you look at the nuclear, there's one species. So these guys are actually still breeding with each other, and yet you do have two distinct nuclear clades showing up in the system. And really not much diversification. We suspect, you know, this is a volcanic lake, and there's probably been an eruption in the past little while that's caused some kind of biological disaster in the system overall. Moving down to Lake Pozo, basically we find one zooplankton, one basic nuclear genome, and the CO1 basically tends to agree with that. Basically a lot of genetic diversity within the population overall, reflecting the age of the lake, you know, system over, you know, as a system. And moving down to the Malili Lakes, it got quite interesting because we basically, you know, kind of a diverse nuclear genome, but still basically one nuclear type seemed to have been the key invader in terms of all three lakes there. But when you look at it in terms of the CO1, you can see some very distinct divergence. Matter of fact, you know, it looked like basically these are from Tuuti, these are Mahalona, Matano, very distinct. But these guys flow into that system. They're zooplankton. You know, Ralph can tell you what would happen to the zooplankton in the water column. It just goes straight down to the next lake, right, Ralph? That's exactly, they can't swim back or they can't do anything else. They're going in there and they're not doing anything. The propical pressure is not resulting in these new clades being able to establish themselves in Lake Tuuti overall. And we think this is basically a result of genetic incompatibility, which has developed, and I suspect that's because the lakes have been separated at one time, and there's been enough differences occurring in these populations that we're not getting the two clades to interact with each other. But as you can hopefully see here, it's evolution in action at all different levels, whether you're interested in fish, interested in snails, interested in zooplankton, interested in phytoplankton. You know, this is one of the best laboratories, as we call it, like Wallace's dream pond, because we're really seeing this modification from the ancestral form And each time is being driven by resource partitioning, resulting in definite tracks of parallel evolution in the terms of structures that are being developed in the different lakes overall. And again, give an idea of the age of these lakes, a lot of genetic diversity in both Tuwuti and Lake Matano. Why are the lakes so unique? I'm going to skip through this quickly. Just very high iron. It's an ultra-mafic area, so that could be it. Fernando said there's high chrome 6 and said that's why we found no cladocerans, but we actually did find a cladoceran last year, but in an area where stream flowed in and it was a little bit nutrient rich. And so perhaps where there is some food, some resources, other species can be existing, and we can actually have some coexistence occurring. Maybe deep adaptations because of a lack of drying events. Another neat thing about this lake, is especially Matano, is this thermal structure here. Top 100 meters behaves like a typical tropical lake, monomictic tropical lake. But below 100 meters, we get basically a meromictic structure. It's thermogenic type of meromixes, and it really results in some really neat chemical layering in the system. I'm just going to skip through there. Looking at iron concentrations, magnesium concentrations as you enter that meromictic thermal structure. High iron, almost no sulfur, highly stable environment. What does that remind you of? The Archean Sea. This is basically what we have found there now. Our photoferrotrophs are living in this system. Bacteria that were found on this planet only billions of years ago have only been ever seen, developed in test tubes before, but basically they're living quite happily in this very unique environment down between about 100 to 110 to 115 meters in Lake Matano. So basically we have a nice Archean Sea relic study system right within our own lake, just sitting there to be worked on. So future research we hope to get into is, again, why no cosmopolitan species, biodiversity, a lot of, Indonesia has like 5 large lakes. Climate change means irregular rainfall. And they've been depending on rivers and constant rainfall. These large lakes are going to become more and more important to maintaining a population of 250 million people. And they are starting, of course, we talk about Indonesia and transmigration is moving people towards the large lakes. So we can sort of see a major biodiversity, a cultural, ethic issue being developed in terms of protecting these types of ecosystems in the longer term. Now, just to finish it off, I think these lakes are going to become the most important lakes in the world. And basically, this is because of what we refer to here as our warm belts in the world, in terms of where the most water moisture is put up into the atmosphere. And that tends to be the Congo, there's the Pacific warm pool, and of course there's the Amazon River. But the big driver, of course, is the Pacific warm pool. Of the El Nino events, all our climate events, all our climate models basically are driven off this system. And of course the variability of the system. And you think about, well, what knowledge, long historic knowledge do we have of this system? Well, there's where we drilled. There's where our deep cores have been. All kind of doing things by proxy, trying to guess what's happening within this area right here. And, of course, and this is, I say, a very important area, but look, Lake Tuwuti sits right dead center in the Pacific warm pool, right dead center in the system overall. In the Pliocene, we basically think in this area there was a permanent El Nino event occurring, very little sort of variability in the system. So it gives you an idea that this Pacific warm pool is really critical to driving our climate. Even the climate here in Canada, of course, is dependent upon variability of the Southern Oscillation, the Southern Ocean Oscillation process overall. So here's Lake Tuwuti's sediments, and I think Ralph probably just opened his eyes because, as an old sediment person, he would love this. This is Lake Tuwuti. These are the seismic chirp lines we took with Jim Russell. This is where we had a fight last March as to where we were going to core. We were going to get 3 sites, but I got one of my sites. I'm happy. But anyway, this is basically looking at the seismics of the lake overall. And you see these beautiful lacustrine structures through the system. And basically we have here roughly is about 150 150 to 160 to 170 meters of beautiful structured sediments. We go back 750,000 years in history, right within Pacific Whirlpool overall in Lake Tawuti. All that has been recorded in these sediments, because sediments are wonderful memories. And as I get to learn this field, like the stuff Ralph knew many years ago, like waxes and these, because that's what he worked on, was chlorophyll and all these paleopigments, this type of stuff. But, we can take a core, and we have taken a core. This is that's a 10-meter core. And I'm just going to show you why it looks exciting right now. That's basically the same thing. Oh, I should tell you, these bands in here is tephra. These are volcanic ash layers. So we actually have from the biologist's point of view, if you're interested, we actually have an endemic system where you look at a community, an event occurs, and then the community reestablishes itself. Oh, and you get to repeat it again. And by the way, you get to repeat it again. Isn't that going to be a fantastic study on basically perturbation and biological recovery of communities? And it's all going to be recorded in a 750,000 year period overall. Like I say, this is going to be the exciting lake of the future, without any doubt at all. Looking at that little 10-meter core we got, you can basically see our ice age through the magnetic susceptibility, which actually is tracked within the iron, so we're actually seeing runoff, shoreline effects. So we know this is working. We know this system is actually tracking these types of events overall, and also in terms of the clastic type sediments in the system. So really, my prediction is probably within about 10 years, everybody's going to be hearing about Lake Tawuti. And guess what? On last Friday, we got told by the Intercontinental Drilling Program they're going to give us $1.2 million to actually go drill the lake. The National Science Foundation came up right away and said, we're in. The German Foundation jumped up and said, we're in. The Japanese Foundation jumped up and said, we're in. NSERC said, we're not interested. That's the world we live in Canada. The major driver of climate change, and we don't have a research program for Canadian science. So if you guys are interested, you're more than welcome to join and come in and work with us on doing these cores. But our first challenge is to go to Ottawa and slap a head someplace and sort of say, it's important for Canadian science to work at the international level. We need international programs within our NSERC system overall. So basically I have to say thank you, but I want to thank some people, especially Peter Hayanus and Didi Hartoto. They were my colleagues in Indonesia, best friends, but both have died in the past year, and and by different reasons and this type of thing. But it's really difficult to work with people that have actually kept me alive, living in rice huts and doing all these types of things and keeping me away from crocodiles. And the strange thing is I'm alive and they're not, and it's the wrong way around. People like Dahlin basically did a lot of our field work for us. Lily Lubis works for Valley. With Inco, she did a lot of the logistics. Graduate students, Dennis Roy, Liz Savo, Andy Bramberger, Ryan Walter, James Biont. They were graduates, and a lot of the research you saw there today was all done by them. By colleagues like Dan Heath, Melania Cretescu, Fabian Herder, Thomas von Rittelen in Germany, Risti Maroto were key collaborators. A lot of the data sets I presented also were from Fabian and Thomas. We do work very well together. It's a really nice international team, really working in this system overall. So at that point I'll say thank you, and I'll take any questions regarding this system or NSERC.
I'm going to